Good morning, class. Welcome to our laboratory session for today. I will be discussing about water analysis for the detection of pathogenic bacteria. Water is very essential to life, so as much as possible, it should not only be safe but also be pleasant to drink. It should be clear, colorless, and devoid of disagreeable taste or smell. And most importantly, class, it should be free from microorganisms. So a wide number of pathogens can be transmitted in by your contaminated water. So it is essential to know whether the water is free of such pathogens or not. And that is a purpose of water analysis. So for today, I will be discussing the common methods for detecting pathogenic bacteria in water. Again, water is essential to life, therefore a good supply must be available to all. Improving access to a safe drinking water can result in good benefit to health, and every effort should be made to achieve a drinking water quality as safe as possible. Those at greatest risk for waterborne diseases are the infants, young children, debilitated individuals, and the elderly. So in general terms, the greatest microbial risks are associated with ingestion of water that is contaminated with human or animal feces. Feces can be a source of pathogenic bacteria, viruses, protozoa, and helminths. For microbial water quality, verification is likely to include microbiological testing. In most cases, it will involve the analysis of fecal indicator microorganisms, but in some circumstances, it may also include assessment of specific pathogen densities. Verification for microbial quality of water must be designed to ensure the best possible change of detecting contamination. The more frequently the water is examined for fecal indicators, the more likely it is that contamination is detected. Testing drinking water for specific pathogens is impractical and can be unreliable. It is neither physically nor economically feasible to test for all pathogens that may be present in drinking water. For this reason, class, tests are carried out for bacteria which are present in feces and indicate contamination of drinking water. These are known as your indicator organisms. These are usually common intestinal commensal bacteria, which are universally present in and excreted in large numbers in human and animal feces. Fecal indicator bacteria should fulfill certain criteria to give meaningful results. So, they should be universally present in high numbers in the feces of humans and other warm-blooded animals. They should be readily detectable by simple methods and should not grow in natural water. The indicator organism of choice for fecal pollution class is your E. coli. Thermotolerant coliforms can also be used as an alternative. So ideally class, water intended for human consumption should contain no indicator organisms. In the majority of cases, monitoring for indicator bacteria provides a high degree of safety because of their large numbers in polluted waters. So class, the most commonly used indicator organisms are the coliforms. So there are tests that can detect coliforms in general, and there are also tests made which can detect specific coliforms such as your E. coli. So it depends on the kind of test. So again, the coliforms class are bacteria that occurs in large number in sewage and feces. They can also be found in the environment in the absence of fecal contamination. So again, just a short review, your coliforms are members of your Enterobacteriaceae. So their general characteristics include they are aerobic or facultative anaerobes, they are gram-negative bacilli, they are non-spore forming, they're able to ferment lactose, and they can produce hydrogen sulfide. So again, like I said earlier, there are tests that can also detect specific coliforms such as E. coli. However, while E. coli is a useful indicator, it has also its limitations. Enteric viruses and protozoa are more resistant to this infection, so consequently, absence of E. coli will not rule out these pathogenic organisms. So in some circumstances, class, it may be desirable to include more resistant microorganisms such as bacteriophages and bacterial spores because these organisms can also cause waterborne diseases. E. coli is a member of the family Enterobacteriaceae, and it is characterized by the presence of the enzyme beta-galactosidase and beta-glucuronidase. It is able to ferment lactose and mannitol and is able to produce acid and gas and produces indole from tryptophan. E. coli class is abundant in human and animal feces, so it is found in sewage, treated effluents, and all natural waters and soils which are subjected to recent fecal contamination, so whether it's from humans, wild animals, or agricultural activity. Therefore, there are water analysis techniques which are used to detect specific coliforms such as your E. coli. Thermotolerant coliform bacteria are those organisms that are able to ferment lactose at 44 to 45 degrees Celsius. So the group includes the genus Escherichia and some species of Klebsiella, Enterobacter, and Citrobacter. Thermotolerant coliforms other than E. coli may also originate from organically enriched water such as industrial effluents or from decaying plant materials and soil. In most circumstances class, the concentrations of thermotolerant coliforms are directly related to E. coli. Their use in assessing water quality is therefore considered acceptable for routine purposes, but the limitations with regard to specificity should always be borne in mind when the data are interpreted. Fecal streptococci are those streptococci generally present in the feces of humans and animals. All possess the Lancefield group D antigen. Taxonomically classed, they belong to the genera Enterococcus and Streptococcus. Most of these species are of fecal origin and can generally be regarded as specific indicators of human fecal pollution for most practical purposes. So, example of characteristics of these organisms is that they are able to survive at 60 degrees for 30 minutes and can grow at a pH of 9.6 and in the presence of 6.5% sodium chloride. So we also have your sulfite-reducing Clostridia. So these are members of the genus Clostridium, which can reduce sulfite to sulfite. The most important of the group is your Clostridium perfringens. So although it is less numerous in human feces than other indicator organisms, its spore can survive in the environment and treatment processes better than most of the other indicator bacteria class. Now let's discuss the analytical techniques used for water analysis. The principal methods used in the isolation of indicator organisms from water are the membrane filtration technique, the multiple tube technique, and the presence-absence test. Membrane filtration method can be defined as the process of microbiological analysis of water by making the use of a special filter-like membrane to trap microorganisms. This is a very effective method for the isolation and enumeration of microorganisms in the test water sample. So, by knowing the quantity of microbial mass, the quality of water can also be determined. Therefore, the membrane filtration method can check both the quantity of microorganisms present in the water and the quality of water. The main objective of this technique is to determine the presence or absence of a particular coliform group that is commonly present in wastewater and groundwater. The water quality is then analyzed by directly counting the number of colonies through a colony counter. So in this technique class, we have the millipore filter. So this possesses a uniform porosity of a predetermined pore size of 0.45 micrometers. The pores in the membrane filter are sufficiently small enough to trap the different types of microorganisms. Following filtration, the filter disc containing the trapped microorganisms is transferred to a sterile petri dish containing an appropriate growth medium. Because the filter is transferred to the petri dish containing an appropriate growth medium, the bacteria present will acclimatize and eventually it will grow. So The passage of nutrients through the filter during incubation will facilitate the growth of organisms in the form of colonies. So the colonies class will appear as clumps. So each of these clumps or particles is therefore called a colony forming unit, and the results are expressed as colony forming units per unit volume of water. So discrete colonies thus formed can be easily transferred to a conformation media. So following incubation, the colonies present on the filter are counted with the aid of a microscope. So again, class, this is an image that shows the setup of your membrane filtration method. So to perform this technique, there's a filtration unit, which commonly is referred to as your membrane filtration assembly. So a filtration unit comprises the funnel, the locking ring, the carbon disc, and the vacuum pump. So there are also many parts that are present. So first, the water sample will pass through the funnel, and a membrane filter is placed over the carbon disc. So on the other end of the filter is a flask. So there is a connector which connects the vacuum motor with the filtration unit. So this vacuum class will provide a negative pressure which permits the suction of filtrate through the membrane filter. So the membrane is transferred to a petri dish with the appropriate growth medium, and the passage of nutrients allows the growth of microorganisms and results in the formation of discrete colonies. The colonies class can then be further transferred to a conformation media or selective media to confirm the type of organism present in the water. So in this step class, the petri dish is incubated for 24 to 48 hours at 35 to 37 degrees Celsius. And when there is growth of colonies, these are then counted using a colony counter with the aid of a microscope. The next technique we're going to discuss is the multiple tube method. So this is also referred to as the most probable number method, because unlike the membrane filtration technique, this is based on an indirect assessment of microbial density in the water sample by reference to a statistical table to determine the most probable number of microorganisms present in the original sample. A most probable number is a statistical method in which the results are compared with the standard statistical table. So this involves three sets of dilutions containing a fermentative broth and the water sample. So this technique class involves three consecutive tests. So you have the presumptive test, the confirmatory test, and the completed test. In the presumptive test, this would involve a series of test tubes which contains the broth in where the water sample is added. Therefore, a positive presumptive test class is indicated by the formation of acid and gas. The quantitative analysis of coliform is determined by counting the number of tubes, giving a positive result, and by comparing the pattern of positive results with the statistical data. The presumptive test can be defined as a screening test which involves a series of fermentation tubes which contain lactose of known concentration. So these are lactose broth class. So as you can see, you have three sets. In the first set, 10 ml of water sample is added, and in the last set, you have your 0.1 ml water added. The test tubes class have a known concentration of your McConkey broth. So a negative presumptive analysis will indicate that the water is microbiologically safe and does not require further testing. But when the results are positive, then further testing is needed to confirm the presence of coliform. The color change of your McConkey broth from red to yellow, along with the presence of gas formation in the Durham tube, will indicate a positive presumptive test. If there's no color change class and there's no gas formation, then that is a negative presumptive test. Again, the multiple tube method class is also called your most probable number method. So this is a statistical method in where the results are compared with the standard statistical table. So as you can see in this image, you have a profile of 3 positive in the 10 ml sample, 1 positive in the 1 ml sample, and no positive in the 0.1 ml sample. So you have a profile of 310. And if you look it in the table, that would give you a microbial density of 43 per 100 ml. So This technique is based on the indirect assessment of microbial density in the water by reference to a statistical table in order to determine the most probable number of microorganisms present in the original sample. After the presumptive test is the confirmed test, and this is used on all positive presumptive test tubes. Sometimes there can be false positive results in the presumptive test, such as your yeast and Clostridium species. So this can be present in the water and can ferment lactose and produce both acid and gas. Therefore, it is necessary to confirm indeed if the growth is from the coliform. So that's where the confirmed stage comes in. So samples from a positive presumptive test tube are transferred to your brilliant green lactose bile broth, agar slant, and your tryptone water. So in the agar slant class, it's inoculated and gram stain preparations made from the slants are examined microscopically. The presence of gas formation in the lactose broth and the demonstration of gram-negative non-spore forming bacilli in the corresponding agar indicates the presence of a member of the coliform group in the water sample examined. The absence of gas formation in lactose broth or the failure to demonstrate gram-negative non-sporing bacilli in the corresponding agar slant indicates a negative test. So that would mean there's absence of coliforms in the tested sample. You also have here class the tryptone water test. So the presence of indole class, as you can see here, there's a reddish layer on top of the layer of the tube. So the presence of indole is indicated by a red color following the addition of your Kovacs reagent. So this forms a film over the aqueous phase of the media. So confirmatory tests for indole, growth, and gas production show the presence of your thermotolerant E. coli because your tryptone water class, this is incubated at 44.5 degrees Celsius. So if it's positive for indole, gas production, and growth, so that would mean that there's presence of your thermotolerant E. coli. If there's only growth and gas production in the absence of indole, this will confirm the presence of your thermotolerant coliforms aside from your E. coli. So after the positive presumptive test and a positive confirmatory test, you now go to the completed test. So the completed test is performed to all samples showing a positive result in the confirmed test. It can also be used as a quality control class. So in here, one or more plates of your eosine methylene blue agar are streaked with a water sample to be analyzed. So after incubation, the colonies are transferred to your tryptose broth and your nutrient agar slant. So again, it's incubated. So from the agar slants class, gram staining is done and it's examined microscopically again. So in the confirmed test class, you can see here the eosine methylene blue agar medium. So EMB is a selective differential agar medium used for the isolation of gram-negative rod. So as you can see here class, there's growth of colonies. So colonies of lactose fermenters will appear very dark purple or have a dark purple center. And a characteristic of the growth of your E. coli is the greenish metallic sheen produced in the growth in your EMB. You can also see here class the formation of gas in the fermentation tubes and the presence of your non-spore forming rod-shaped bacteria from the agar culture. So these results class will is considered as a satisfactorily completed test. The last technique to be discussed is the presence-absence method. So the presence-absence test class is much more appropriate for monitoring good quality drinking water where positive results are known to be rare. So it is not quantitative, and as the name suggests, the method only indicates the presence or absence of your indicator organisms. So in the presence-absence method class, you have a container, usually it's a bottle, with a folded tissue paper inside, and that folded tissue paper will contain aliquots of concentrated media, which contain the ingredients to support and indicate the presence of organism growth. So after that, it's sterilized, and then water is added to the bottle. A positive presence-absence method is manifested class by a change of the color of the water from clear to brown to black. So it will indicate fecal contamination and also indicate the presence of your indicator organism. So what is the principle behind this class? So coliforms have enzymes that are able to produce H2S by reducing sulfur. And the production of H2S class will combine with iron, which is present in the alicot. And usually iron class is in the form of your ferric ammonium citrate. So when it combines with the ferric ammonium citrate, it will form a black precipitate. And that is the reason why there is a color change when there's a positive of your, positive indicator organism present. That ends our lab session for today, class, and I hope that you've learned something about the few techniques of water analysis and its importance in preventing waterborne diseases. So thank you and keep safe.